Hi everyone, I'm Paige Mitchell, and today I'll be discussing my work on characterizing the pelvic and hindflipper muscle anatomy of leatherback sea turtles. Leatherbacks are the largest living turtle species and hold records for their geographic distribution and their diving abilities. The mortality rate of leatherbacks continues to increase due to anthropogenic causes and has led to their classification as being critically endangered. Improving our knowledge on their anatomic structure and function may facilitate our conservation efforts by creating opportunities for advancing medical interventions and, in turn, helping our repopulation efforts. In this study, we decided to have our method sections divided into two: diagram and model creation, and muscle identification and documentation. The two specimens that were used in the study were obtained from the Canadian Wildlife Health Cooperative, and the hind end of each specimen was removed after necropsy and were frozen. The CT and MRI scans were taken, as shown in Figure 4, and the data was then uploaded to 3D Slicer to segment and render the structures into three-dimensional models, as seen in Figures 1 and 2. Once created, the models were then uploaded to Mesh Mixer to fix any errors that were created in the mesh, and were finally uploaded to HalletBox to slice the models for 3D printing, and Fusion 360 to create specimen-specific diagrams to help our dissections. After all of this preliminary work was completed, we were able to start our dissections. Muscles and their associated attachments were identified, described, measured, photographed, and removed, as shown in Figure 3. The tendons were removed from the muscles, and both of them were dried in 60 degrees Celsius, and both the tendons and muscles were then weighed individually. The final results for this study are pending. Quantitative analysis will be performed on the functional and regional groups. The expectation of the results are that the hip flexors and extensors will be significantly lower in mass, and that the distal flipper extensors, flexors, supinators, and pronators will be significantly greater in mass than that of other sea turtle species. We also expect to find that the flexor pubotibialis muscle is present in leatherbacks. This study will continue throughout the next 2 years to increase our sample size and confidence in the results. Having diverged from other sea turtle species 100 to 150 million years ago, it is expected to find that there is appreciable differences in the hind end muscle size, position, and attachment sites from that of other sea turtle species. The number of attachments that were found directly to the dermis suggests that the rudder function of the hind end in leatherbacks is highly utilized and would lead to the increased muscle mass of the associated muscles. The need for detailed diagrams became very evident during the dissection of the right hind limb of the female specimen due to the significant branching patterns of the tendons, which were very challenging to identify accurately. Other challenges did arise, particularly with creating the virtual bone models within 3D Slicer due to differences in tissue densities and noise that was created in high-density areas in the scans. Collectively, these factors led to non-specific tissue selection within the software's tools and required manual correction within each individual slice for anatomic accuracy. So references for this work are found at the bottom of this poster, and my contact information is also found on that page. Please let me know if you ever have any questions, and thank you so much.